Students, in the first video, I explained you about commercial applications of biotechnology, that is, crop biotechnology and animal husbandry. Now let us listen about next applications: human health, white biotechnology, environmental biotechnology, food biotechnology, and DNA fingerprinting. Let us study first about human health. Diagnosis and treatment of the disease are two important aspects of the human health management. Biotechnology helps to identify the role of gene. Diagnosis of diabetes and heart disease has become possible even before the onset of symptoms with the help of biotechnology. Diagnosis of the disease like AIDS, dengue can be done within few minutes. Hence, treatment can be done at the earliest. For example, the hormone insulin is used in treatment of diabetes. Earlier, insulin was being collected from the pancreas of horse. However, nowadays, due to biotechnology, insulin can be prepared with the help of bacteria. For this purpose, human insulin gene has been inserted into the genome of bacteria. Various vaccines and antibiotics are also produced in the same way. Now, let us study about more applications of biotechnology in contact to human health. First is vaccines and vaccination. Students, vaccine is the antigen. Antigen means any substance that can stimulate the production of antibodies and combine specifically with them. And vaccine is the antigen containing material given to acquire either permanent or temporary immunity against a specific pathogen or disease. Traditionally, vaccines were prepared with the help of pathogens. Completely or partially killed pathogens were used as vaccine. However, due to this, there were chances of contracting the disease in case of some persons. Hence, as an alternative, scientists tried to artificially produce vaccines with the help of biotechnology. For this purpose, scientists produced the antigen in laboratory with the help of gene isolated from the pathogen and used it as vaccine. Thus, safe vaccines are being produced. Now, proteins which act as antigen are injected in pure form instead of injecting the killed or partially killed pathogens. These proteins keep the person away from disease by keeping the immune system active. Thus, injecting the antigens is safest way in vaccination. Vaccines produced with the help of biotechnology are more thermostable and remain active for longer duration. For example, vaccines of polio and hepatitis. Students, you will be amazed to know that the day is not far away when, instead of getting drops of injections of vaccines, you will be able to getting immunity by simply eating some vegetables. We may call them edible vaccines. Scientists are developing transgenic potatoes with the help of biotechnology. For this, the desired gene is isolated from human pathogen like Vibrio cholera and E. coli, and it is transferred in transgenic plant virus. Now, this virus allowed to infect fresh potato leaves. These infected leaves are then used to develop entire plant by vegetative reproduction. And potatoes are collected. Consumption of these raw potatoes will generate immunity against cholera and disease caused by E. coli. Students, what will happen if transgenic potatoes are cooked for consumption? Let me tell you the answer. We will not get immunity because if transgenic potatoes produced as edible vaccine are cooked for consumption, heating at high temperature would cause denaturation of the protein and reduce the potential and effectively of the vaccines. Hence, these potatoes need to be consumed raw. Biotechnology is useful for production of hormones and proteins useful for treatment of disease. Hormone insulin is used for treatment of diabetes and hormone somatotropin, also known as growth hormone, is used to treat dwarfism. Students, next is interferon. This is a group of small size protein molecule used in treatment of viral disease. These are produced in blood. Transgenic E. coli are used for production of interferon. Next is gene therapy. Genetic disorders like phenylketonuria, that is PKT, in somatic cells. All the cells except sperm and ova arise due to genetic changes in hepatocytes, that is liver cell. It has become possible to treat it with somatic gene therapy only due to biotechnology. Students, you might have heard the term cloning. Production of replica of any cell or organ or entire organism is called cloning. It is of two types: reproductive cloning and second is therapeutic cloning. Reproductive cloning means a clone can be produced by fusion of a nucleus of somatic cells with the enucleated ovum of anybody. Thus, there is no need of sperm to produce the new organism. Do you know? A sheep was cloned by this technique few years back. Let us peek into history and find out. Peeking into history. A sheep, Dolly, was born in Scotland by cloning technique on 5th July 1996. Nucleus from the udder cell of sheep of Pin Dorset variety had been introduced into enucleated ovum of Scottish sheep. Then the ovum was allowed to develop in the uterus of Scottish sheep and thereby the dolly had been born. It was showing the characters as per the chromosomes in the nucleus, and any character of Scottish sheep was not visible. Next is therapeutic cloning. Stem cells can be derived from the cells formed in laboratory by the union of somatic cell nucleus with the enucleated egg cell. Various diseases can be treated with the help of these stem cells. Similar to cells, genes can also be cloned and millions of copies of same gene can be produced. Those can be used for gene therapy and other purposes. Controlling the inheritance of hereditary disease, continuation of generations, enhancing the specific tendency may become possible due to cloning technique. However, there is worldwide opposition to human cloning on various issues. Next concept is industrial products or white biotechnology. Various industrial chemicals can be produced through less expensive processes by using biotechnology. For example, alcohol production from sugar molasses with the help of transgenic yeast. Next is, let us study about relation between environment and biotechnology. It has become possible to solve environment related various problems with the help of biotechnology. Microbial techniques are already in use for treatment on sewage and solid waste. Sewage is rich in organic matter. If such sewage is released in natural water bodies like rivers, the organic matter in it get oxidized with the help of dissolved oxygen. Due to this, level of dissolved oxygen in water decreases and adversely affecting the aquatic life. As a remedy on this, sewage should be released into rivers only after oxidation with the help of microbial technique. Microbes are useful on large scale, while production of compost by treatment on solid organic waste material. Bioremediation, biopesticides, biofertilizers, biosensors are some new concepts in biotechnological methods. Now let us study about what is bioremediation. It means either absorption or destruction of toxic chemicals and harmful pollutants with the help of plants and microorganisms. If plants are used for this purpose, it is called as phytoremediation. Some examples of bioremediation are as follows. First is the fern Teres vittata can absorb the arsenic from the soil. Genetically modified variety of Indian mustard can absorb selenium from soil. Sunflower can absorb uranium and arsenic. The bacterium Deinococcus radiodurans is highly radiation resistant organism. It has been genetically modified and used to absorb the radiations from radioactive debris. Grasses like alfalfa, clover, and rye are used in phytoremediation. Next is food biotechnology. Food items like bread, cheese, wine, beer, yogurt, vinegar are produced with the help of microorganisms. These food items are probably the oldest one produced with the help of biotechnology. Students, the next term is DNA fingerprinting. DNA, that is deoxyribonucleic acid. DNA fingerprinting is useful in forensic science and paternity testing because the DNA sequence pattern of every individual is unique and hence it is known as the DNA fingerprint of an individual. A person's identity can be established with the help of a DNA sample. At a crime scene, any source of DNA, that is blood, or semen found can be used to establish the identity of the criminal. Also, the paternity testing or the identity of a child's father can be established using DNA fingerprinting. So, to students, white biotechnology, that is industrial products. Industrial biotechnology that employs living cells or microbes to produce industrially useful products through a less expensive process is known as white biotechnology. Next is environmental biotechnology, food biotechnology, and DNA fingerprinting. That's all for today. Thank you.